Deep within a silent laboratory, a researcher stares intently at a glowing screen. It displays the intricate neural map of a mouse brain, a glowing web of infinite complexity. For decades, we believed the brain's journey to maturity was a solo performance by neurons, a simple tightening of electrical connections as we grow. But today, the spotlight shifts. We are looking past the neurons at a hidden orchestrator lurking in the shadows, the star-shaped astrocyte. These cells were once thought to be mere structural support, but they hold the secret to how our minds lose their youthful flexibility and settle into the rigid architecture of adulthood. Inside a controlled chamber, a young mouse opens its eyes for the very first time. As the first rays of light hit the retina, a biological chain reaction begins. A signal surges through the body, triggering a release of corticosterone. This hormone travels like a messenger through the bloodstream, racing toward the brain's visual cortex. But it does not seek out the neurons directly. Instead, it searches for a specific docking station on the surface of the astrocytes, the glucocorticoid receptor. It is a precise lock-and-key mechanism. When the hormone finally clicks into this receptor, it signals the astrocyte to begin a profound physical transformation, forever changing the landscape of the developing brain. Under the influence of the hormone, the astrocyte begins to reshape its very architecture. It secretes specialized proteins that weave together, building a physical cage around the neurons. This structure is known as the perineuronal net. Think of it as a molecular break. This net locks the brain's circuits into a stable, mature state, ensuring that our learned behaviors and sensory perceptions are preserved. While this stability is essential for survival, it comes at a cost. It effectively seals the door on the rapid learning and adaptability of youth, marking the definitive transition from a flexible mind to a permanent, rigid structure. To challenge this biological law, researchers devised a daring experiment. They injected a virus into the visual cortex of adult mice, specifically designed to delete the glucocorticoid receptors within the astrocytes. Weeks later, they performed the ultimate test: monocular deprivation. They covered one eye to see if the adult brain could still rewire itself in response to change. In normal mice, the brain remained rigid and unresponsive, unable to adapt to the loss of sight. But in the modified mice, the results were staggering. The visual cortex shifted and adapted. By removing that single receptor, the molecular break had been released. The neurons began to form entirely new connections, proving that the adult brain is not permanently sealed. It is merely held in place by a latch we have finally learned how to touch. As the data flickers on the screen, the implication is clear. Maturity is not a loss of potential, but a deliberate, hormonal process of stabilization. The door to plasticity is not welded shut. The latch is simply engaged. Within these cellular interactions lies the future of medicine. We are looking at a path to repairing damaged neural circuits, turning the rigid architecture of the adult mind back into a landscape of endless possibility. The mind remains a place of growth, waiting for the right key.